Good morning, everyone. So welcome to our webinar. Today, our topic will be on the 16S gene sequencing, and this is the application of the next generation sequencing. So, we'll go through the next generation sequencing, give a brief introduction first. Then we're talking about later about the 16S rRNA gene sequencing, and then we will discuss a little about the sample preparation and how to do the sample preservation and the DNA isolation. This will given by Norwegian scientist Dr. Kim, and then we continue to sequencing part, and also including data analysis. Then final part we will give some example. So we know that the next generation sequencing, like normal sequencing method, it also go from library preparation, sample handling, and then next is the deep sequencing, and the final step will be the data analysis. So unlike the traditional sequencing method, the single sequencing, the deep sequencing, or called it next generation sequencing, we will do massively parallel sequencing, which means that we will get millions of reads at the same time, and with the parallel sequencing, we have a high throughput for the next next generation sequencing, and it can go to very large. It can go to the very large scalability, and considering how many reads we get in a few days, it's very fast speed. And data analysis is mostly important part now because we have our standard sequencing platform. But in order to get the meaningful biological information, we must have we must do the data analysis. And with this, with those bioinformatics tools, we can derive the information. Derive the biological information from the sequencing reads. So for 16S rRNA gene sequencing, we use the Illuminous MySeq desktop sequencer as our sequencing platform, and the length of reads can go up to 2 multiply 300 base pairs. That's a paired end read, and the maximum number from the MySeq goes to 25 million reads. That's a lot of reads for 16S rRNA sequencing, gene sequencing. So the data output will 15 gigabases. The runtime for the whole paired end 2 multiply 300 base pair length will be 65 hours. 65 hours. That's probably close to 3 days. So we know that for the 16S rRNA sequencing, we just sequence one target applicant from the bacteria or microbiome. So for example, before we normally use a single sequencing method, which is just isolate one microorganism from the mixture, from the soil, this kind of thing, and then sequence one colony after we did the culture. And we sequence like single sequence 550 base pair, or even goes to 900 base pair. But we kind of limited our sequencing rate to one or maybe 96 wells. 96 tubes from this. But for the Illumina's MySeq system, we can get like 25 million reads. So if we calculate this, that's probably we can handling a lot of samples. So once we want to mix the samples together, we need to have some way to do the multiplexing. So the next one I show how we handle, how Illumina's protocol handle this kind of situation. So we put multiple samples together, sequencing parallelly, but we need to have a barcode. So the barcode is both at the 5' end and the 3' end, and the multiple sample mixed with this barcodes. And after parallel sequencing, we get multiple reads, and based on the barcodes will assign the reads to different sample according to previous assignment of the barcode. And then after getting the read, we assign the reads, and then we can get the reads based on the barcode and have them sorted together into one sample, another sample. So this one, by this method, we can, by this barcoding method, we can have multiple samples sequenced at one time and analysis later of the multiplexing. So talking about the 16 rRNA gene sequencing, we know that we are looking at the bacteria mixture or maybe microbiome. It's not a single colony anymore. So the sample is come from environmental samples, or maybe come from the gut microbiome, and also we normally want to check what's the contamination in the food and what's the infection from our body. So we also check this kind of samples. But here again, we must keep in mind that this is all mixture of the bacteria. It's not a single colony anymore. So we put a few literature here to demonstrate what are this 16S rRNA gene sequencing applications. So later you can take the slide and go for the detail. So interestingly, in 2011, the literature for the 16S rRNA gene sequencing is about 600, at more than 600 papers. And after the first use of next generation sequencing for 16S rRNA gene sequencing, in about 4 years, the publication almost doubled. So now in 2014, the projected number for this application were about 1,200. So metagenomic sequencing is not the new topic, and before we always do the isolate the microorganism first and make it grow in the autoclave or some other culture medium, and then we purify DNA, extract the DNA from the isolated bacteria, and then sequence the whole genome by single sequencing or later by the next generation sequencing. And this is still working on the single colonies or maybe single isolated one. And then there comes another metagenomic sequencing that they don't isolate the colony or separate the bacteria anymore. They just sequence the whole DNA extracted from the mixture, bacterial mixture, or from the soil, from the water, something, and then sequence all the fragments from the multiple microorganism. And after that, assign the reads based on the alignment to know what species, what's the taxonomy inside that mixture. And for the 16S rRNA gene sequencing, we are not going to sequence the whole genome from each species. So we just sequence the applicant from target on the 16S rRNA gene. And based on the variable region of the 16S rRNA gene, we know and we follow the alignment to the database of the 16S rRNA gene. We know what the species inside, what's the genus of this all the microorganism inside that sample. So we call this method is 16S rRNA gene sequencing. So because we just look at one applicant, and so the identification job, task will be much easier and efficient compared to the traditional identification method to sequence the whole genome from each species in that mixture. So also we have a very good database for the 16S rRNA gene, and so we can do this job more quickly and accurately. And we know that the 16S rRNA gene do have a multiple variable region, like here in the in colored one, and between them there's a constant region, like this. Among them is constant region. So we designed the sequencing primer, the PCR primer, based on the sequence from the constant region, and then fish out the variable region. For our protocol, we just target on the V3 and the V4 region, and then this dual zone detection, which can be let us give them obtain the sequence longer sequence and make it a more accurate analysis of the colonies inside the mixture. And also we just sequence the one applicant and have a very deep coverage, very high coverage. So the cost will be low, and also due to the deep sequencing, due to the high coverage, so we have the high sensitivity, which even the low abundant bacteria can be identified. So give a flowchart for this 16S RNA sequencing. We go like this. So first we harvest the bacterial or population from the skin, for example. So we extract the DNA from the skin sample, and then simply just use a one-step PCR. We know that there's a few protocols talking about the two-step PCR. First, get the applicant, and then put the barcode at both ends. So, but our protocol is just as simple as one-step PCR. We already, the PCR primer already contains barcode and also sequencing primer region. And then only one single PCR step, we captured the 16S V3 and V4 region, and then we sequence this after mix multiple samples together. And based on the barcode, we do the multiplexing and then assign the reads to different samples, and then do the alignment of the sequences reads. And after alignment, we know which one, which species, which read is from which species. then we can do a good taxonomic classification, use the current available reference database on the 16S rRNA gene. And after that, we can do the final genetic analysis to see the evolution trees like this, and to know the relationship between the mixture of the bacteria inside that sample. Here, the case is the skin samples. So now the first step for all the biology work will be the sample collection and extraction. I will give this to our Norwegian scientist, and he will give us more details about the sample extraction, DNA extraction.
Good afternoon. Hi, everyone. I'm Won-Shik Kim, a senior research scientist from Norwich Biotech. I'm happy to share some few aspects related with the DNA extraction for environmental samples, but the microbiome and food assurance today. The bottom of the successful 16S DNA sequencing, one of the important prerequisites is the high quality of the DNA extraction. Having said that, this fact hasn't been changed for many decades, explaining how the DNA extraction has become a fundamental and critical job, despite the fact that more and more sophisticated downstream applications are exponentially available nowadays. Here are common samples where most of the metagenomics studies have been conducted, and I would like to share some of the challenging issues for DNA extraction from each samples. So, soil case, most people are very well aware of that. The humic acid is one of the biggest problems when they isolate DNA. It's certainly the level of the humic acid inhibits PCR or any other downstream applications. The ocean, including salt or fresh water, usually contain a different level of ions that also inhibits kind of the DNA purification, particularly a column-based purification system, and depends on the water sample, particularly from the liver and the sludge in it. The humic acid could be done because of the poor DNA purification. And more importantly, the low DNA is the kind of key that how to really concentrate the DNA from the vast amount of the water samples. For the food assurance, there are a variety of the food. There's a fresh or a processed food. Many processed food case preservatives and also starch or starch-coated fat can be a kind of hurdle for the clean DNA purification. In the milk case, many human pathogens somehow can be present in the milk, so people are quite interested in to detect and understand the microbiome in the milk. However, the fat, naturally generated fat, actually somehow also became a hurdle for the DNA extraction. There are two other bodily fluid, which is urine on the top part, and then also fecal samples at the bottom. These are really one of the difficult samples to handle. However, due to the increasing amount of the interest to understand the microbiota in the fecal samples, there are a lot of the demand to get the high quality DNA. But again, a lot of the processed byproduct in the fecal samples, as well as the humic acid level, actually make the DNA isolation difficult. The urine case, it naturally contains a high level of urea and a salt, And more importantly, the low DNA amount also quite difficult to get a good amount of DNA for a sufficient PCR or sequencing analysis. So speaking of the DNA isolation, however, there are not such universal DNA isolation method available. Thus, it is important to find the right DNA isolation method. So good DNA extraction kit for samples we mentioned before should deliver at least a wide range of the sample acceptability. That includes all the challenging environmental samples what I just mentioned, and also should really deliver clean DNA which has a really great ability to remove the PCR or any sequencing inhibitors, and also should really deliver sufficient DNA yield, and more importantly, the rapid and also user-friendly performance. Another aspect I would like to bring up here is the importance of the sample collection and preservation. 16S rRNA DNA sequencing is significantly influenced by the population of each microorganism group in the sample. If the population is affected by the sample collection or transportation method, which is sensitively affected by temperature or duration of the transportation time, the result of what you will have will be quite far from the original sample. So in order to prevent or minimize this oddity, the original sample can be preserved chemically using preservatives. The benefits are you can ship the samples at room temperature, and also to preserve or sterilize the intact biological information from the site of the collection to the facility for DNA extraction. And it will be a convenient method that you don't really need to have a bulky ice pack or cooler, and then it's more cost effective for the long distance shipping. Here are selected a case study recently published by users using Northern products, and a few cases will be introduced by Dr. Su in this presentation later. So just in brief, there's soil and then water and then milk, and the bacterial genomic kits has been used for network topology, distinct microbiome community study, and also the raw goat milk microbiota, and so on. So I'd like to turn over to Dr. Zhu for the rest of the presentation, and I'd like to thank you very much for your time. Please let us know if you have any questions. I and Lisa will be available to the end of the webinar and happy to answer your questions.
Okay, thank you, Dr. King. So we continue our work. Once we have the good DNA sample, so we will do the QC first, and then if the DNA positive QC, we go further to the PCR step. So as I mentioned, that this is a one-step PCR instead of two steps. So we are looking at the applicant region V3 and V4 variable region of the 16S rRNA gene, and it's about 470 decibels. So I mentioned that my signal can go up to 2 multiplied 300 per the end, so it exactly covers this about 470 decibels, and we can have a good data. And the primary information here listed here is that what the PCR primer we are using. The census strain, forward strain is called a 319A, and the reverse strain antisense PCR primer is called a 806R. And based on how many samples we want to sequence in one MySQL run, we need to put the 5x. It's about 10% to 70% based on the number of the samples. And the purpose of the 5x is increase the sequencing complexity to make the MySQL sequencer recognize the reads, recognize the sequencing rate. And start sample material here, I want to emphasize is that they are the DNA samples, not the RNA. Even we are looking at the 16S rRNA, but we are looking at the 16S rRNA gene. It's DNA. It's not RNA sample. And sample requirement, we would like to have 2 micrograms for each sample, and the concentration should be more than 10 nanogram per microliter. And we know that it's just a PCR reaction here to amplify the amplicon. But if we have a good quantity of DNA, that will be much easier to do the sequencing job instead of using the low quantity DNA samples. So here is the data analysis part of our sequencing work. So first, that once we get the raw data from the sequencer, we will, based on the barcode and also linker primer, we will assign the reads to different samples, as I mentioned. And then we will clean the data to remove some low quantity reads. And then the good thing after that is we do the OTC cluster. So all these steps, even the data, intermediate data, will also provide to our customer. For example, like this panel shows, we have raw data and also have the clean data, and we will have the OTU profiling data for each samples. And OTU cluster, if I make it simple, is that just after alignment, we put the unique reads together. So we know that from each bacteria, we may have one candle of 16S fragment, amplicon fragment. So based on that, we can do the taxonomy assignments to see, based on the database, the known database, 16S rRNA database, we know what are these reads coming from, which species, which genus. And then if we have multiple samples, we can do the OTU profiling. We can have the hidden map. Because sometimes we know that some sample is traded, or some sample is from different environment or different condition. So we want to compare the different situation to get the biological information from that. And then with the heat map, we can have a better idea to immediately visualizing to see what's the difference among these samples. And certainly we can also have some species classification. And with that information, we can do the diversity analysis, like alpha or beta diversity. And with the beta diversity analysis, we can also do the principal coordinate analysis. And finally, we get the RACI-5 OTU table. So all this information, data set will be sent to the customer, sorted, organized like the right panels. And here, so I want to show one example of our data analysis summary report in our data analysis sample report. First panel, we will give the information about the sample, like what kind of sample type, and the receiving date, and what we did, and what's the project ID. And we also provide the 16S being a primary information. And the database we used will be listed in the database information, like the standard one, the very popular one, green gene, and its link, and also RDP, ribosomal database project, and their link. And the customer is also welcome to provide their own database if they have some special requests. And after sequencing, we will know how many reads we get for each sample, like the table listed here. And after we clean the data, assign the reads, we know each sample still keep how many reads left, and then we know how many reads for each sample, and we know they are still fit to our standard. Normally, we want to provide a minimum 100K reads for each sample. And the taxonomy community distribution is the most important part. We want to see what is the taxonomy from our sample. And the pie chart is shown here is for the final of this of the sequence, the sample. So we know the distribution, we know the abundance for each silo, and also we provide the pen chart based on the class, and also provide the pen chart for genus. So from this pen chart, we know the distribution of the microorganism, and we know the abundance of them. And this is the bar chart. So this one will clearly show the also show the distribution of the microbiome, and what are these sign-on for the text assignment, and for each samples, and also based on the class. And the heat map I just mentioned that this is giving the customer the genus heat map. So for each sample, once we compare among the samples, we know which genus is highly abundant in which samples, or maybe it's lower abundance in different samples. By this method, we know if the sample is from different condition, or with the treated or untreated condition, we can do a good comparison among them, and make a call to see which microorganism is highly abundant inside, and then we know the profiling of all these microorganisms inside the samples. And certainly we also provide a long table and a long list on what all the genus, species, not species, genus inside our sequenced samples. So this side is a taxonomy. It shows what's the sign-on, what's the class, and what's the genus of the sample we identified, from the sample we identified. And the count is just the reads, how many reads we found from this genus, and what's the percentile inside of the sample sequence. And this is the total, and we also give the numbers for each sample. And then from this long list, we know how many genus we identified from the sample, and what's the abundance, what's the distribution there looks like. So we have a lot of tables on different sample, and also combine the comparison, the comparison between their samples and between the taxonomy communities. So we have tables on genus and synonyms, and also different classes. Okay, we go through the data analysis a little bit, and I can provide a few cases. And this one, the authors want to take a look at the analysis on the raw goat milk microbiota. And so they have two kinds of milk. So one is the wild type, another is transgenic goats, which can naturally take the lysozyme inside the milk. And so they want to see if after the transgenic code express the lysozyme in their milk, which can prevent the growing of the bacteria or not over time. So they did the 16S. They extracted the DNA using the Nogent's kit. You know, want to emphasize that they did use the Nogent's kit to extract the DNA. And interestingly, they found out that even the milk contained the lysozyme inside. They didn't show much difference of the microorganism pattern among this between the wild type and also the lysozyme expressed milk. And even during the early stage and the middle stage and also later stage, they have a similar pattern. They compared the distribution side by side. So that's their conclusions that of the sequencing comparison with the sequencing data. Their conclusion is that the presence of the lysozyme did later to influence the microbiota of the goat milk. Another case is that scientists want to see the buried soils in the Siberia tundra area. So they know that in this this area is also the soils keep frozen and keep frozen in this area. They want to dig out what's the microbiome community structure in this soil, including the topsoil and also the buried soil. And the interesting finding is that in the topsoil, they see a lot of fungi species inside. And the buried soil, they found out that because of this anoxia condition and also the very low temperature, they did find that the fungi was not highly abundant. But the bacteria took place, the fungi's abundance. I mean, they become abundant, and they are functionally well to decompose the soil organic matters inside. So they use the 16S method to find out the distribution of the different microorganisms inside the soil. And this case is more clinical related. So we know that in the swimming pool and other place, there's a lot of aerial monos, which may cause some disease to the human. And so in this case, they want to identify what exactly the aerial mono species inside. And so they use multiple methods, including 16S rRNA gene sequencing. So they successfully identified 94 species from the aerial monos. So here they also emphasize that based on one method, you cannot get a very good clear picture of the microorganism inside. So you may need to use multiple methods like GCAT and also target sequencing method on the RPOD genes. So we conclude our webinar. So today we discussed a little about the 16S rRNA gene sequencing using the Illumina's MySeq platform. Our company is headquartered in Houston, Texas, and we provide the service including later sequencing service since 2005. Now we already handled more than 10,000 samples, and our customer is around the world, and it's almost from 1,400 institutes to college and hospitals. So it's all from 40 countries. And thank you, and you're welcome to send us questions. You can write your question now, or maybe later you want to ask a question and give us feedback. And please email us at the info@lcsciences.com. Thank you so much.